Hi, so in today's video I'll be looking at photosynthesis. It's a process where energy from light is used to break apart the strong bonds in water molecules, and the hydrogen is combined with carbon dioxide to form glucose, and oxygen is released into the atmosphere. This happens in chloroplasts. These are organelles that are found in plants, and they capture energy from the sun, and then they convert this into chemical energy that is stored either as sugars or other organic molecules. Photosynthesis nourishes the entire living world, so directly or indirectly. Some terms we need to know before we go on. The first one is autotrophs, so these are self-feeders. Which means that they don't consume other living organisms. So they produce their own organic molecules from carbon dioxide and other inorganic raw materials, produce all the organic compounds that is needed for heterotrophs, which we'll discuss shortly. So autotrophs are usually plants. We've got heterotrophs, we cannot make our own food, so we have to consume compounds produced by others. And we are known as the consumers, so we eat plants and animals to get our energy. So this is the main equation for photosynthesis. So we take 6 molecules of carbon dioxide plus 6 molecules of water to produce 1 molecule of glucose and 6 molecules of oxygen. So glucose is the ultimate energy source, and you actually obtain the energy in form of ATP through a process called respiration. I'll do a video on respiration shortly. Respiration comes in 2 forms, either aerobic or anaerobic. In aerobic, we use oxygen. In anaerobic, there is no need for oxygen. Why do we need energy? So in humans, things like muscle contractions, homeostasis, active transport, DNA replication, cell division, and protein synthesis, and the same goes for plants. So let's look at photosynthesis in more detail. So there are 2 main stages. Stage 1 is known as the light-dependent reaction, which happens in the thylakoid membranes of chloroplasts, and in this process we're converting solar energy to chemical. So here is an image of a chloroplast, and the light reaction happens in these stacks, which are called thylakoid membranes. And the second stage, which is the light-independent reaction, also known as the Calvin cycle, which happens in the stroma, which is the liquid inside the chloroplasts, and this is when you incorporate carbon dioxide into organic molecules, which then allows us to convert it to simple sugars. So this is the Calvin cycle shown here. Phosphorylation is when we add a phosphate group to a molecule. So for example, ADP is phosphorylated to ATP. Photophosphorylation is when we're adding a phosphate group using light. Photolysis is the splitting, so lysis means splitting of a molecule using light energy. Hydrolysis is the splitting of a molecule using water. So for example, ATP is hydrolyzed to ADP. And finally, a redox reaction is where we have oxidation and reduction taking place at the same time. So if something is being reduced, it gains an electron. If something is being oxidized, it loses an electron. So oxidation of one molecule always involves the reduction of another. An easy way to remember is to remember oil rig, which stands for oxidation is loss, reduction is gain. So chloroplasts are small flattened organelles which are found in plant cells. In the green parts of the plant, so mostly leaves, which are the sites of photosynthesis. They have double membranes called the chloroplast envelope, and we find them in the mesophyll layer, so shown here on the leaf, and this is the cross-section of the leaf. So these are the chloroplasts, and this is a mesophyll cell, and inside a mesophyll cell we've got these organelles, chloroplasts. So carbon dioxide enters and oxygen leaves through micropores called stomata. Inside the envelope is a fluid called stroma, and in the stroma we have the sacs called thylakoids, and they're often stacked and as shown here. So if we have many of them, we call them grana, or a single one will be called granum. All these grana are interconnected and have molecular machinery that carries out photosynthesis. Chloroplasts also contain photosynthetic pigments, so chlorophyll A, chlorophyll B, and carotene. And these are colored substances that absorb the light energy, which is needed for photosynthesis. And the pigments are found in thylakoid membranes, and they're often attached to proteins. And when they're attached to proteins, we refer to them as a photosystem. We need to know that there are two photosystems, photosystem 1 and photosystem 2. Photosystem 1 absorbs light best at a wavelength of 700 nanometers, and photosystem 2 at 680 nanometers. A dependent reaction, we need light. It takes place in the thylakoid membranes of the chloroplasts. And here the light energy is absorbed by the photosynthetic pigments in the photosystems and then converted into chemical energy. So in the light-dependent reactions, ATP is made by photophosphorylation. So we can either make ATP from ADP, an inorganic phosphate. This is what is referred to as photophosphorylation. We're making reduced NADP from NADP. And we're also splitting water into protons, electrons, and oxygen, and this is called photolysis. So here I have a diagram of what the first stage looks like, and I'm just going to walk through it quickly. So first of all, we'll start off on the left here. So the light energy is absorbed by photosystem 2, as represented here. The light energy excites electrons in chlorophyll, and the electrons move to a higher energy level. Then these high energy electrons move along the electrons transport chain to photosystem 1. So these green ones represent chlorophyll. So as the light strikes them, they get excited, and an electron then goes into the primary acceptor, which then gets passed on along the electron transport chain to photosystem 1. As the excited electrons from chlorophyll leave the photosystem 2 to move along the electron transport chain, they must be replaced. So light energy splits water into protons, electrons, and oxygen, which are shown here, to replace the electron that's leaving. And you can write it as H2O going to 2H pluses plus half of O2. The excited electrons lose energy as they move along the electron's transport chain. So this energy is used to transport protons into the thylakoid, so that the thylakoid has a higher concentration of protons than the stroma. This forms a proton gradient across the membrane. So this is shown here. And then as protons move down their concentration gradient into the stroma via an enzyme called ATPase synthase, the energy from the movement combines ADP and inorganic phosphate to form ATP. And this is shown here. So light energy is absorbed by photosystem 1, again shown here, which excites the electrons again to an even higher energy level, and finally these electrons are transferred to NADP along the proton from the stroma to form reduced NADP, as shown here. This is known as the linear electron flow, so non-cyclic photophosphorylation. There is also cyclic photophosphorylation, but in this process only ATP is produced, and it only uses photosystem 1, and it's called cyclic because the electrons from the chlorophyll molecules aren't passed on onto NADP, but they are passed back to photosystem 1, as shown here. So they're basically recycled. So it just keeps going through photosystem 1 rather than continuing to photosystem 2. And this process doesn't produce reduced NADP or oxygen. It only produces small amounts of ATP. So let's look at the light-dependent reaction, also known as the Calvin cycle. This happens in the stroma of chloroplasts. Carbon enters as carbon dioxide and leaves as a sugar. So the cycle consumes ATP and NADPH. So ATP is used as the energy source and NADPH as the reducing power. Stage 1 is known as carbon fixation. We'll start at the top here. So CO2 enters the leaf through the stomata and diffuses into the stroma of the chloroplast. Here it's combined with ribulose biphosphate, which is known as RUBP for short, which is a 5-carbon compound. But this gives a very unstable 6-carbon compound, which quickly breaks down into 2 molecules of a 3-carbon compound, which is called glycerate 3-phosphate, or short G3P. Rubisco, which is an enzyme, catalyzes this reaction between carbon dioxide and ribulose biphosphate. So this is the first stage. The second stage is when ATP is reduced to NADP, as required for the reduction of G3P to triose phosphate. So now ATP from the light-dependent reaction, as shown here, provides energy to reduce the 3-carbon compound G3P to triose phosphate, or also known as glyceraldehyde 3-phosphate. And this reaction requires H pluses, which come from the reduced NADP, which also comes from the light-dependent reaction. The reduced NADP is recycled to NADP. The final stage is when ribulose biphosphate is regenerated. So 5 out of every 6 molecules of triose phosphate that are produced in the cycle aren't used to make hexose sugars, but are used to regenerate RUBP. So the regeneration uses the rest of the ATP which is produced in the light-dependent reaction. So to produce 1 GA3P, the Calvin cycle needs to consume 9 molecules of ATP and 6 molecules of NADPH. So then when the cycle turns 3 times, we get 6 molecules of GA3P, as 2 of them are made for every CO2. So 5 out of the 6 are then used to regenerate RUBP. So for 3 turns of the cycle, only 1 GA3P is made. However, glucose is a 6-carbon sugar, so we need 2 of the glyceraldehyde 3-phosphate to make 1 glucose. So the cycle therefore must turn 6 times to produce 2 of GA3P, which will then give us 1 glucose. So when the cycle turns 6 times, it uses up 18 molecules of ATP and 12 molecules of reduced NADP. The Calvin cycle is the starting point for making all the organic substances a plant needs. So the triose phosphate and glycerate 3-phosphate molecules are used to make carbohydrate, lipids, proteins, and nucleic acids. So carbohydrates, as we know, are simple sugars, for example glucose, and they're made by joining 2 triose phosphate molecules together. Or if you want to make a polysaccharide, so for example starch or cellulose, we need to join hexose sugar together, and we can produce different polysaccharides depending on how we join the hexose sugars. We can also produce lipids, and these are made using glycerol, which is synthesized from triose phosphate and fatty acids, and fatty acids are synthesized from glycerate 3-phosphate. We can also make amino acids, so some amino acids are made from glycerate 3-phosphate, and finally nucleic acids. So the sugar in RNA is ribose, which is made from triose phosphate. And finally, just to relate how the structure of a chloroplast is adapted for photosynthesis. So the envelope keeps the reactants very close to the reaction site. The thylakoids have a very large surface area, which allows as much light energy to be absorbed as possible. We also have many ATPase synthase molecules present to produce ATP in the light-dependent reaction, and the stroma has all the enzymes, sugar, and organic acids present so that the light-independent reaction can take place. So I hope to finish off, we would look at some past papers. First of all, let's look at this question from June 2011. The diagram here is showing the double membrane envelope of a chloroplast, and the first question is asking us to complete the diagram to show the structures involved in the light-dependent reactions of photosynthesis, and we're also asked to label these structures. So, since there are 2 marks, we first need to draw the grana, the stacks of thylakoids. So first of all, you would draw maybe 2 or 3 of the stacks, and also make sure you label them. So granum, which is a singular, and if you have many, just grana, and also make sure you mention the thylakoids. So that would be 2 marks. Then the table below shows 2 statements taken from a student's essay about the light-dependent reactions. Complete the following table by placing a tick in the correct column next to each statement to show whether it is true or false. So the first statement says electrons and chlorophyll are excited as light energy is absorbed. Well, this is true because once they're excited, they then can go through the electron transport chain. So this statement is true. The next statement, the energy absorbed by chlorophyll is used to generate ADP and NADP. That is not true. And the final question is asking us to explain how oxygen is produced during the light-dependent reactions of photosynthesis. So first, there are 2 marks, so we need to mention first of all that get the energy from the light, so from the sun. We then need to mention the process of photolysis, which is the breaking of molecules using the light as the energy. And obviously, to produce oxygen, we need to break a molecule of water. Photosynthesis can be divided into 2 main stages, the light-dependent stage and the light-independent stage. Explain why the light-independent stage cannot take place without the light-dependent stage. So we have three marks, so we have to make sure that we at least mention 3 points. The products from the light-dependent stage are needed for the Calvin cycle. The products of the light-dependent stage are reduced NADP and ATP. Reduced NADP is used for reduction of carbon dioxide and GP, and we also use ATP as the energy source for the Calvin cycle. So these are the things we need to mention. And another question from January 2012. Place a cross in the box that identifies each of the following. So substance W, which is here. So we asked to say, could it be carbon dioxide, an electron, an inorganic phosphate, or a proton? So it definitely cannot be B or D. So it can either be carbon dioxide or inorganic phosphate. Well, in this case, it's inorganic phosphate. Then for reaction S, which is the arrow going forward, is it either carboxylation, hydrolysis, phosphorylation, or photolysis? Well, it's actually B, hydrolysis, because we're breaking up H2O into protons, electrons, and oxygen. And finally, reaction T, which is the arrow going back to ATP, is as carboxylation, hydrolysis, phosphorylation, or photolysis. And in this case is phosphorylation because we're adding a phosphate to produce ATP. So to B, reaction T occurs in a chloroplast. Describe the structures in chloroplasts that are involved in this reaction. So again, we have 3 marks. We have to make sure that we make at least 3 points. So first need to refer to the grana, which is where photosynthesis takes place. Granum is a stack of thylakoid membranes, and they are connected. Thylakoids contain electron carriers. They contain the photosynthetic pigments chlorophyll, which forms the photosystems. So photosystem 1 and photosystem 2. Electron carriers and ATPase are associated with the thylakoids. So they're the ones that form the electron transport chain. And the final question is the energy released by reaction S is used to form GALP, glyceraldehyde 3-phosphate, during the Calvin cycle. Plant cell walls contain cellulose molecules. Suggest how GALP may be used to synthesize cellulose. So this has got 5 marks. Glyceraldehyde 3-phosphate is a 3-carbon molecule, and we can use 2 of those to form glucose or a hexose sugar, so a 6-carbon sugar. In the synthesis of glucose, enzymes are involved which help with the reaction. To remember that cellulose consists of beta-glucose, which is a 6-carbon sugar, which is joined by glycosidic bonds, and the bonds are 1 to 4. So to form cellulose, we need to go through a condensation reaction between glucoses, and cellulose is a long chain molecule, so it's a polysaccharide. So it means we're simply joining many monomers together to produce a polysaccharide, and it's unbranched. I hope you found this video useful. Thank you very much.